Special thanks to Ziv and the scientific committee and the other organizers of the meeting. This is becoming my favorite meeting every year, so very excited to be back. And I'm going to talk about a case. It's an ordinary case, ordinary in the sense that it's a very typical presentation of Parkinson's disease. Unilateral onset of resting tremor in an individual in their 60s, levodopa responsive. But at the same time, this case is exceptional because I think it demonstrates what we can learn when we dive very deeply into a single case and try to understand the drivers. So as you can see here, this individual was diagnosed with GBA Parkinson's disease. He has a positive family history, has a T408M GBA variant, which is one of the two GBA variants that increase risk for Parkinson's disease, but do not cause Gaucher disease. And this variant is present at about, in the population at 0.01 and causes about an increased risk of Parkinson's disease about 2 times. It's reducing GCase activity by about 20%. And you can see that the variant is also shared by 2 siblings who are not known to have Parkinson's disease. I can tell you that this individual, because we've sequenced the whole genome, has a low to average polygenic risk score, which we've found in this publication that's being cut off here. First author is Shaw. We found that low polygenic risk score can actually be a predictor for GBA Parkinson's disease, that it can indicate the presence of a less common or rare variant cause. So this variant falls on an allelic spectrum of genotype, phenotype, with regard to penetrance of GBA Parkinson's disease, as shown in this nice figure from a study that Clement Scherzer led. And you can see here the really nice relationship between the effect of the variant on GCase activity and the penetrance for risk of Parkinson's disease. And I, like Tony, am a believer in the loss of function model because of data like this. So what I'm going to focus on today are the questions that I'm highlighting here. One, what are the cellular consequences of GBA partial loss of function? And is this sufficient to trigger nervous system dysfunction? So what is going on in carriers of GBA? And 2, do other lysosomal storage disorder genes influence PD risk, and how? And I just call your attention to the left side of the plot where we have non-GBA PD, and you can see there's really a remarkable range of GCase activity measured in individuals without GBA variants. There may be many reasons for this, but could there be other genetic variants that are affecting lysosomal function that possibly interact with GBA to affect and modify penetrance? So we got into this going back almost 10 years now in a study that Tony very generously mentioned during his talk. So in this study, we looked across 54 lysosomal storage disorder genes, and we asked whether, as a group, those genes increase risk for Parkinson's disease. It was a 2-stage design, and I show you some of the details here. And what we found is that indeed there is a rare variant burden for predicted damaging variants across this whole interesting group of genes that cause lysosomal storage disorders. We could remove GBA from the analysis, and the risk remained, suggesting that there are other non-GBA genes there. And over the years, indeed, through larger studies, some of which I show here, a number of other lysosomal storage disorder genes have been proven to be PD risk genes, including SMPD1, RSA, GALC. And so the way I've been thinking about this as a kind of hypothetical frame or model is that if you nearly completely lose function of one of these genes, you develop often a pediatric lysosomal storage disorder. But if you have partial loss of function, even modest partial loss of function, in combination with aging, you may be at increased risk for developing Parkinson's disease. And the question, which I think Tony really nicely kind of posed to us all, is what is the mechanism of this? One potential mechanism is increased risk for alpha-synuclein-related neurodegeneration, because the lysosome is an important organelle for the turnover and proteostasis of alpha-synuclein. So in order to look at this hypothesis, we've actually systematically looked at lysosomal storage disorder genes and their ability, when knocked down, to modify alpha-synuclein-induced neurodegeneration. This was published a couple of years ago, so I'm just going to present a highlight or two. But in this study, we did a genetic screen in a Drosophila fruit fly model of Parkinson's disease, in which alpha-synuclein is expressed all throughout the nervous system. And we looked at more than 90 different conserved Drosophila homologs of lysosomal storage disorder genes. Some human genes have more than one homolog, which is how you get to 94 from the 54 that I mentioned. We tested more than 300 different lines in this screen. And I'm showing you one example for the gene NPC1 in flies, which is the homolog that is conserved of human NPC1 that is a cause of Niemann-Pick type C, and NPC1 encodes a cholesterol efflux transporter from the lysosome. And so you can see in this plot on the left, we use locomotor behavior climbing as a screening assay. In black is the climbing of flies as we age them, control flies. You could see that when we remove 50% of NPC1 activity, there's no effect on climbing, that's in green. Synuclein expression across the nervous system in flies causes this progressive locomotor impairment. And when we add 50% reduction in NPC1 in that context of synuclein, we can see this strong worsening climbing phenotype. And then in some cases, we go back and we look for neurodegeneration, as I'm showing you one example here, where you can see this vacuolar degenerative change that we can assay in the Drosophila model is strongly enhanced. So it turns out that not only NPC1, but 15 in total Drosophila homologs of lysosomal storage disorder genes interact with alpha-synuclein, and we can categorize them into genes that have an additive interaction and those that have a synergistic interaction. Additive means that when we remove function of that gene on its own, it affects locomotor behavior. And this is not unexpected because many of these lysosomal storage disorders, in fact, will affect nervous system function. And many of these genes over the years, including some from our own work and others from the literature, have now been implicated in PD risk. And our study also uncovered GBA itself. And in this table, I'm organizing the genes based on the metabolic pathways that they affect. So in this study, this study is consistent with the hypothesis that other lysosomal storage disorder genes can modify alpha-synuclein-induced neurodegeneration. But what about modifying GBA itself? So going back to the 2017 study, what we found, which was really intriguing at the time, was we were looking at more than 1,000 exomes when we did our lysosomal storage disorder burden analysis. And about 20% of the cohort had actually 2 or more variants in lysosomal storage disorder genes. And so the suggestion of this to me at the time was the possibility that an individual could have multiple hits within this interesting gene network. And kind of a little bit like the way we currently think of cancer, where you need multiple hits to ultimately develop the disease, it could be that GBA, in combination with other lysosomal storage disorder genetic variants, could push the lysosome into a dysfunctional state that leads to Parkinson's disease. So we've tried to look at this, and it's been 10 years. This is not an easy thing to look at. And I don't think we finished yet, but first of all, we've looked at it both with human genetics and fly genetics, and I'll kind of take you through a little bit of each. So on the human genetics side, I'm showing you some unpublished work from Jeff Kim, who is a postdoc in our group, and he's here at the meeting. And what Jeff did was to, in human genetics, He did a rare variant genetic association analysis comparing GBA PD cases to all other PD cases, meaning PD without GBA. We designed the study this way because we were mostly using GP2 data for this analysis, and there aren't very many GBA non-manifesting carriers in that cohort. So this was the most sensible way to do this analysis. And we first looked across this interesting gene set of lysosomal storage disorder genes. And what we found is that there is a burden of damaging variants across the lysosomal storage disorder genes that seem to characterize GBA PD when compared to all other PD, consistent with the hypothesis of oligogenic modification. We then did a gene-by-gene analysis across this gene set, and we found a couple of interesting hits, but far and away the driver of the burden signal was a variant in NPC2. And NPC2 is another cause of Niemann-Pick type C, another involved in cholesterol efflux. This is a really interesting variant. Firstly, it's one that's present in 1% of the population. The variant is a predicted splicing variant. It is not a Niemann-Pick C causing variant. This variant has a large odds ratio for an effect on GBA penetrance. It is a strong effect, and importantly, we think this may be the first pure modifier of GBA penetrance because this variant or NPC2 are not known to affect PD risk. And of course, we've looked hard in all the available human genetic data. So this is a variant that can modify GBA penetrance, but doesn't seem to independently cause PD. And what's more, this variant, while I mentioned it's not pathogenic for NPC, the variant is a functional variant because it is a strong effect on NPC2 protein levels in blood based on data from the UK Biobank. It reduces NPC2 protein by about 40% on its own in heterozygosity. The association we found here replicates in the UK Biobank, and so we think it is quite solid. But human genetics can only take us so far, and so we've gone back to the fly to look at this more systematically. And so just as a preface to this part, I want to point out that this is work that was done by Jinghan Zhao, and she has a poster that will show some of this later today or tomorrow. Actually, I can't remember when you have your poster, Jinghan. And I also wanted to mention that both of these studies were funded in part by the Silverstein Foundation for Parkinson's disease. So Jinghan spent a long time looking at whether GBA heterozygous flies with a 50% reduction in GBA have any nervous system phenotype. She was mostly disappointed by this data, but it's actually the expectation because many of us walk around with GBA, partial loss of function, and we're not aware of any neurological consequences to it. And so what I'm showing you here is that GBA in heterozygosity has no effect on fly locomotion, even when you age flies, has no effect on neurophysiology when we do electroretinograms from the fly eye, and then we've even looked at this ultrastructurally by aging animals and doing transmission electron microscopy. Here I'm showing you the retina. So GBA partial loss of function, we can't find anything in a fly phenotypically. What we then did is we took these flies, GBA heterozygous flies, and we now crossed them to each of the lysosomal storage disorder gene that I talked about earlier in heterozygosity. So this experiment, to be clear, is done without synuclein present. We're just looking for interactions between GBA and other lysosomal storage disorder genes in double heterozygosity. And we've looked at more than 30 different conserved genes, including all of the ones that we looked at in our prior study. And the most interesting interaction was the fly homolog of ATP13A2, which causes a rare, atypical Parkinsonism with dystonia, but it's a recessive disease. Here we're looking at a double heterozygous interaction between GBA and ATP13A2. There are a couple of other genes that appeared to interact or worsen GBA in heterozygosity, but those genes gave phenotypes on their own in heterozygosity. So, but the fly ATP13A2 homolog, as you can see in this data, gave a clear synergistic interaction, and it wasn't just climbing. This work was done collaboratively with my close colleague Hugo Bellin at Baylor College of Medicine, and the interaction is seen for neurophysiology, as shown in the data here. We also aged animals, and you can see on transmission electron microscopy that double heterozygosity causes this retinal degeneration phenotype. And metabolically, we see elevated glucosyl-ceramide and some other ceramide and sphingolipid species. So GBA and ATP13A2 interact in double heterozygous animals. And what's more, and I don't have time to go into all the details here, but in a twist, it's looking like this interaction is occurring non-cell autonomously. GBA is mostly functioning in glia in flies, and ATP13A2 is mostly functioning in neurons. And as far as the mechanism goes, our best bet, based on a lot of data that's in our now published paper and an earlier paper, is that ATP13A2 has an important role in the exocytosis of glucosyl-ceramide. So in the presence of neuronal activity, there we see glucosyl-ceramide accumulating. The neuron exocytoses glucosyl-ceramide, the glia takes it up, and GBA is functioning in glia to break down glucosyl-ceramide, and ATP13A2 may affect that exocytic process. So you may have seen this coming, but going back to the case that I started with, it turns out that besides the GBA variant, this index case has variants in both ATP13A2 and NPC2. The ATP13A2 variant is strongly predicted to be damaging, and we've looked at large databases and found what appears to be a 7-fold enrichment for damaging ATP13A2 variants among GBA PD cases when compared to GBA carriers without PD. So it's consistent with the idea that these variants might modify GBA penetrance, but these variants are very rare, and I don't even want to give you a p-value because it's not quite proper to compute one, but the data is consistent. We've also found an NPC2 variant. It's not the one that I told you about, but this variant is also a variant that reduces NPC2 protein levels in UK Biobank data, so it appears to be functional. But you can see here that even with GBA and these two other variants, we can't fully explain PD in this family because there is a sibling that has all three as well. And so what I haven't looked at yet, of course, are common genetic variants. We've been focused on rare variants. There are many common genetic variants affecting lysosomal genes or lysosomal storage disorder genes. We focused on one. I probably can go quickly through this part because I spoke about this variant last year at this meeting. But this is a variant that affects an interesting gene called SPTSSB. It is a genome-wide significant common variant. This variant is associated with a highly significant expression quantitative trait locus in the brain that increases expression of this particular gene. This is a regulatory subunit for an enzyme SPT that initiates ceramide synthesis. This is happening in the ER. And SPTSSB is thought to alter substrate specificity, and there's some really nice mouse work suggesting that it can increase sphingolipid base length and promote neurodegeneration. And I want to say it's really important to look at the functions of these variants in healthy control individuals. Because if you're looking at PD cases, you are confounded by the PD pathophysiological process and the effects of synuclein. We found that this variant increases sphingolipids and ceramides, and you can see it here in plasma from healthy controls. And we have this causal chain between the variant and PD risk. So it turns out that our index case is homozygous for this variant, whereas the two siblings are heterozygous. And so in the final minute here, I just want to talk a little bit then about this hypothesis, about how these variants interact. The hypothesis is that GBA1 partial loss of function causes a subtle subclinical signature of lysosomal or metabolic perturbation. And combinatorially with other variants, this might push the lysosome to be dysfunctional and could push your way towards Parkinson's. So we've been looking for this subclinical signature. In order to do that, and I know I'm gonna, like, final 10 seconds here, we've done multi-omics of our GBA heterozygous flies, going back to the fly. What I'm showing you here is that in RNA sequencing, you can see in a three-way PCA plot that when we look at all the transcriptional changes, age can separate the three groups, and GBA heterozygosity is distinct from controls, and it's intermediate between a wild-type control and a complete GBA loss of function. There's an overlap, as I'm showing in the Venn diagram, between the RNA, the dysregulation of expressed genes in GBA heterozygous and homozygous flies. And what's more, we found a very interesting signature of altered lipogenesis. And so what you can see here is that several different genes that encode enzymes important in lipogenesis are partially reduced in GBA heterozygosity, intermediate between wild-type controls and GBA homozygous animals. And all of these genes are responsive to the SREBP transcription factor, which is a master regulator of lipogenesis. And so we think, and we've been working collaboratively with Tawfiq Raj, and we think we have some evidence that replicates this finding in human monocytes from non-manifesting GBA carriers. So, of course, we've looked at the metabolome as well, and there are a lot of metabolic changes, and you can visit Jinghan's poster, and hopefully she'll explain them all to you. But there are changes in triglycerides that are reduced, consistent with fatty acid lipogenesis defects in GBA heterozygous flies. And maybe I'll regret putting this on here, but I can't find other examples, but maybe if there is an example, someone in this room is gonna come tell me during the question period. I can't find other examples of endophenotypes in GBA heterozygous carriers, but I think that understanding what happens in GBA carriers before they develop Parkinson's disease will be really important to understand how GBA Parkinson's disease happens. So my final slide is just simply to say that in this particular case, there are multiple hits within a metabolic network that seem to be converging on lysosomal function and sphingolipid metabolism. And that's the hypothesis that we're now pursuing. And I tried to thank everyone who contributed to the work along the way, and sorry to go 2 minutes over. Thank you.